So today I'll shift gears from chloride to calcium to sodium. I wish I can borrow your slide. Sodium is the most important ion. So, and I'll tell you about this darling of the sodium channel field, the sodium channel 1.7, which has sort of become the bastard stepchild. And hopefully, I'll show you that it still remains a darling of drug targeting for chronic pain. So it's backwards, you said, right? All right. So very quickly, it is turning out to be there's like 7 talks over the next 2 days on pain. So ion channels are very important for pain. I'll tell you very quickly that pain is a global phenomenon here in the U.S. We have a lot of people suffering from pain, high-impact chronic pain. About 100 million people suffer from this. And it has a very great degree of mortality that's associated with chronic pain. Chronic pain is also encumbered by other things like sleep disruption and depression and anxiety. So it really is a problem that is not going away, and it's been exacerbated by the opioid crisis. And pain, to give you past these numbers, pain is really driven by both central and peripheral mechanisms. And today's talk will focus really on a peripheral mechanism revolving around the voltage-gated sodium channel NAV 1.7. And it's been said that persistent pain may be simply driven by events that start in the peripheral nervous system. And I realize this is a very channel-centric crowd, so this next slide is probably going to be very obvious to most of you. But nevertheless, I'll tell you that NAV 1.7 is a very critical channel that drives the upstroke of the action potential. Look at it a different way. Here's Jimi Hendrix playing his guitar with his teeth. Next to him is a big, huge amplifier. The amplifier is 1.8. But nothing's coming out of the amplifier unless you have something driven by 1.7. It really is the critical channel for driving up action potentials. And much like as Diane was telling you, location is very important. This channel is present in the right location in the skin nerve endings, in the dorsal ganglia sensory nociceptors, as well as in the spinal cord. And really the claim to fame and why it became a darling target was about 20, 25 years ago when it was discovered that mutations in this channel cause these really weird phenomena of gain of function, which is a lot of channel activity, so these very painful disease scenarios, and conversely, mutations that cause congenital insensitivity of pain. So both these kind of mutations really galvanize the field towards targeting this particular channel. But as I'll tell you, this channel has really been very hard to drug, not least of it because of the fact that there's other members of the family. And there have been strategies to develop differentiated approaches, but they haven't really been successful. Another reason for the failure to achieve analgesia is the fact that we don't really know in humans how much block is needed to achieve perfect analgesia without compensating or without jeopardizing the protective block. Another reason for the failure is this issue of CNS versus PNS. This is really not going to be solved anytime soon, more than likely. And it's unsure really where the block of 1.7 or some of these channel targeting drugs has to be. And if you do cross over the blood-brain barrier, are you then going to be encumbered by side effects? And finally, a new twist to this story has been this emerging story in the last 5 to 7 years that shows that much like calcium, sodium is acting like a second messenger. And the data shown here is a little bit controversial, but says that if you delete 1.7, in other words, you need to really completely block with whatever drugs you're targeting to drive an upregulation of endogenous opioids. And failure to do this is why some of the drugs that are in the market are not going through to clinical use. And of course, many other things that plague the pain field, we could be doing better biomarkers, better trials, and perhaps using some of the newer human iPS cells or human cells to de-risk some of the compound strategy development. So with that in mind, I've shown you what the problem is. We turn to ask the question, could we target this channel in a differentiated manner from what all the pharma and other folks have done? And we did this by looking for the molecular composition of this particular protein and how some of these players dictated the function of this protein. And in this context, we discovered a particular protein, which I'll tell you about now, which is CRIM2, collapsin response mediated protein. This protein was found to be a key player in driving the function and activity of this particular channel. And it really begs the question that we really have to look at the channels not in isolation, but really study the interaction platform for these particular channels. This protein was really discovered in the context of its ability to drive axon growth. And in fact, in response to semafrin, it collapsed. So that's the name, collapsin response mediated protein. My lab's been working on this for a few years now, and we discovered a connection to this protein in terms of neuroprotection through NMDA receptors and a sodium-calcium exchanger. But the story today I'll tell you really gravitates towards how CRIM2 can drive the trafficking of NaV1.7 and how we can harness that to develop a small molecule, like a tool that Merit was talking about, which now we're trying to get to the preclinical stage. So let me try to convince you that this 1.7 is regulated by this particular protein. So CRIM2, we discovered, interacts with 1.7. And to test whether it's a functional interaction, we knocked out CRIM2 with an siRNA from DRGs and looked at sensitive currents. This is 1.7, TTX sensitive currents. And you can see a decrease in the channel density. This was fully recovered if you put in a knockdown resistant version of CRIM2. So in other words, CRIM2 was really responsible for executing this decremented current. And here's the knockdown confirmation. The TTX resistant currents were not affected. CRIM2, as I said, is a pleiotropic functional protein. It executes all these different quadrants of functions. So how does this really control 1.7 specifically? Well, looking carefully at the structure of CRIM2 and its sequence, there are a lot of hotspots for phosphorylation, for modifications. And my lab discovered a modification called SUMO. It's like a ubiquitin, small ubiquitin-like modifier. And this protein is modified by SUMO. And what we discovered really was that if you simply delete or prevent SUMOylation from happening with proteases that cleave SUMO, you now see the same decrease in current as you saw with the loss of CRIM2 completely. So this gave us first proof that simply SUMOylation was enough to recapitulate the decrease in 1.7. Looking at this further, we mapped out exactly where SUMOylation was happening. And among the various lysines that could serve as a candidate acceptor site for SUMOylation, lysine 374 happened to be the lysine that contributed to the regulation of NaV1.7. To convince you that this is happening in real neurons, in nociceptors, we examined DRGs that have been transfected with wild-type CRIM2 or a null form of CRIM2 that wasn't SUMOlatable. And as you can see, the 1.8, 1.9 resistant currents are not affected. The TTX-sensitive currents are indeed decreased, but they're composed of these particular channels. If we take out of the equation from these 1.7 with Protox, now you see no difference. So the inference is that all of this is coming because of an effect on 1.7. And we looked at 1.5 and 1.3, and none of those were affected. And this is really because of trafficking. As I failed to show you, on the right side, you have this annulus expression of 1.7 on the surface in wild-type CRIM2 expressing cells, but in the cells that have a mutant form, you don't get the channel to the surface. And it's because the channel is trapped in these early endosome markers. If you don't believe the confocal data, we approach this in a functional manner because some of these are involved in clathrin-mediated endocytosis. So what we did essentially is look at expression of these currents in wild type compared to K374, which are decreased. But if you put in pit stop, which blocks the endocytosis pathway, now you see a full recovery of the current. So it's really a trafficking effect imposed by loss of CRIM2 stimulation on 1.7. With this data in hand, we proceeded to make a knock-in mouse that recapitulated the 374 phenotype. And here is exactly what we did. We made a CRIM2 K374 homozygous mouse, knock-in mouse, and very gratifyingly, it also showed the same phenotype in that the currents were reduced from DRGs with this sumo null mutations knock-in. And consistently with the previous data, it is a surface trafficking effect of sumo loss of stimulation on the channel. And very importantly, because it's a sort of a pain-centric talk, and we're trying to figure out how this is contributing to pain. If you were to inflict a model of injury in these mice, the 334 mice, in this case with a spare nerve injury in both females and males, you see the wild type are very sensitive to paw withdrawal thresholds, as Dan said earlier, as a measure of tactile hypersensitivity. But amazingly, the female CRIM2 knock-in mice and the male knock-in mice didn't first fully develop the same pain thresholds as the wild type, but they quickly recovered their pain from the pain in this model. So this really hit homes the message that simply loss of CRIM2 stimulation is enough to drive the trafficking of the channel to a lower degree. So you have less excitability, and in an animal model, now you see this recapitulated and manifest as a loss of pain, so refractory to pain. So based on this, several years ago when I was in Arizona, we started a company called Regulonics, regulators of ion channels, sort of a portmanteau of those two words, to really address this emerging and unfortunately continuing global health epidemic, particularly more so in the US. And where most people have been doing this kind of a strategy to block the channel, which is useful, but it's going to lead to absence of complete sensation and lack of selectivity, the Regulonics approach has really been to understand, as I said, the molecular dialogue, which led us to discover the simulation of CRIM2 driving 1.7, and really looking at enzymes, for example, UBC9, which brings sumo to CRIM2, to see if we could design small molecules to then revert this phenotype to this and create a less painful situation. So this work started with my wife's group, who is a crystallographer. Her group solved the crystal structure of CRIM2, shown here, and we started on a small high throughput screen where we docked molecules onto the simulation site of CRIM2. Two hotspots emerged, and here you can see the CRIM2 protein sitting here. Here, this is UBC9, which brings sumo onto CRIM2, but sitting right smack in the middle is this green molecule called 194, which is preventing access for simulation of CRIM2. So this is the molecule that we basically, after several rounds of SAR, were able to optimize. And for the next few slides, I'll tell you about this molecule and try to convince you that this is really sort of harnessing the same mechanism as I've told you in the past few slides. So what does 194 do? Remember, 194 is designed to block the simulation of CRIM2, which would then execute this series of events to internalize the channel and have less sodium currents. First, we verified that the molecule only binds to CRIM2. It doesn't bind to a related CRIM5 or to UBC9. UBC9 is lethal, embryonic lethal, so we don't want to touch it because it also targets other proteins. We then found that UBC9 interaction with CRIM2 can be reversed or blocked by 194. And finally, simulation, shown here, can be also inhibited in vitro by 194 in a manner similar to the lysine mutant, if you will, genetic mutant. So this is probably the most important slide because it goes to show the selectivity through this differentiated mechanism. So it's a busy slide, but it's fairly simple. In small DRGs, we recorded with DMSO these robust currents for sodium. With 194, we can see the channels decreased. Protox, which is a sort of a fingerprint toxin for this channel, also decreases 1.7 currents. But if you combine them together, you don't see any further decrement, which means that this decrease is entirely due to 1.7. We verified this was in small DRGs. In large DRGs, you don't see that effect, so it's specific to 1.7. And then we scanned through all the sodium channels and found that only 1.7 was affected. CAV2.2, the N-type channel that Diane talked about, is not affected by this particular compound. A compound in our hands gave an IC50 of 1 micromolar. In an independent replication study in Tim Huchel's lab in Cologne, Germany, it gave an IC50 of 2 micromolar. As a way to move this forward, de-risk this in other species, we tested the compound in mouse. It also inhibited in mouse DRGs, inhibited in pig DRGs, and also in human DRGs. But the final proof that this mechanism was really because of CRIM2 stimulation came because of the fact that we had the CRIM2 null mice. And in this experiment, what we did essentially is look at wild-type mice. As I told you earlier here, for example, we can block 1.7 with 194, shown here. The sumo null mice already have reduced currents, as I had shown you before. But very importantly, when we put in these DRGs from these mice, 194, you have no effect. So really, 194 is selective. It works through a CRIM2 stimulation-dependent mechanism. And consistent with the fact that it has an effect on trafficking, we looked at assessing trafficking 2 different ways, using confocal microscopy here, and you can see a very clear difference, as well as cell surface biotinylation. And again, this protein, 1.7 was affected, the trafficking in 1.7 was affected. Another protein, CDK5, for example, was not affected, nor stimulation was changed. Mechanistically, because 194 is preventing the stimulation, now the complex that starts to nucleate the process of endocytosis is now free, and it can be more associated with the channel to internalize the channel itself. So that's really how mechanistically this 194 compound is working. Looking at it from the perspective of endocytosis and clathrin-mediated endocytosis, as I showed you earlier, we did the same experiment with looking at recordings in DMSO. We know 194 blocks are 1.7. If you put pit stop with these experiments simultaneously, you now see that you can recover the current, which again suggests that the mechanism involves internalization and endocytosis of this particular channel. This has been independently validated in several labs, in the labs at the NCATS, the National Center for Advancing Translational Studies at NIH. And they have replicated it in human-derived nociceptors, red DRGs, a mouse cell line, as well as in Al George's lab in Chicago, who's looked at this compound and seen very robust replication of our data. So in the last few slides, I'll tell you, I'll show you a little bit about moving this in vivo and show you some behavioral effects. So far I've shown you in vitro. What about target engagement in vivo? Well, indeed, if you give animals 194 and then look at and prepare spinal cord lysate, you can see stimulation is affected by 194, as it should be doing. But this is sort of the primary the indirect target, but the primary target is 1.7, remember? So we looked at looking at fractionation of pre and post density of the spinal cord, and this is vehicle injected versus 194, and you can see a clear difference. This is the first demonstration that this is really working in vivo to traffic or prevent the trafficking of this channel. All right, moving on to the pièce de résistance, if you will, for looking at pain. So we, of course, evaluated this molecule in various pain models. Here's a couple of examples. This is the model of spare nerve injury. And here you can see the compound reverses tactile hypersensitivity. In this model, orally, in a spare nerve ligation model, you can see dose-dependent oral relief of pain. We've tested it in a bevy of other models, including some neuropathic models, chemotherapy-induced peripheral neuropathy. We've tested it in formalin in a post-surgical model, as well as an itch model, and it's successful in all of these models. It doesn't have any development of tolerance, so here we did, again, an SNL model, and then delivered 194 systemically through an LZ pump, so a little bit of compound is being delivered over time. And as you can see, there's a little bit of loss of efficacy, but when the pump stops, you see it coming off. So during the time, there's really no development or loss of tolerance of this compound. One very important thing that could emerge should this compound get to the clinic could be its synergy with other existing analgesics. We all know, unfortunately, that opioids are never going away. And so here we tried to ask the question, could our compound synergize with some of the classically available analgesics? And here is, for example, showing you that a subtherapeutic dose of morphine, which has no side effects, also has no beneficial effects, if combined with the subtherapeutic dose of our compound 194, now shows you pain relief. So this could be somewhat of a strategy to lower the side effects of opioids, if you will. Finally, I'll tell you, we scoured the other behaviors to see if anything else was affected by 194. Locomotion is not affected. Behavior for exploration, anxiety, depression is not affected. 17 is very, very important for your sense of smell, which is also not compromised by our compound. It's been really fortuitous that this program was taken up by NCATS about 2 years ago, and they have done something that we couldn't do with their 25 medicinal chemists. They've taken our compound 194 and really evolved it through a very systematic SAR approach. And what I will tell you is that for example, where our compound initially, 194, was about 15 to 20 minutes half-life, the compound that we have now as our clinical lead is 20 hours. So that's one of the parameters they've really optimized. And so this is our lead compound that's modeled on 194. It has excellent bioavailability. The half-life is phenomenal. And it has somewhat of a brain penetration, but we also have compounds that can be segregated to just peripheral versus just central. So I think we're now at the precipice of taking this really to the clinical stage, and we're now trying to talk with companies to move this forward. So in summary, what I've shown you is that by understanding a novel mechanism involving CRIM2 stimulation, and rather preventing CRIM2 stimulation, we are able to develop a compound that can block UBC's delivery of SUMO to CRIM2. This compound relieves pain orally. It reduces NaV1 point currents specifically in various species, including humans. It's not addictive. This is data I didn't show you. It's not addictive, and it may synergize or potentiate the activity of other analgesics. So I think we're onto something here with this new mechanism of stimulation to drive trafficking of 17. With that, I'll tell you that this is the new group at NYU, and this is a long-time collaboration with my wife's group. And finally, I want to thank the funding from NIDA and NIH, and a grant from SCTR, as well as the disclosure that I have Regulonics. Happy to take any questions.
Expect any effects on the other functions of CRIM2? You showed the wheel of different functions the molecule has when you modulate the stimulation.
Great questions, thank you. So we have looked at 194 in cultured neurons, and it does drive up endogenous opioids a lot more than some of the compounds that were shown in some of the previous studies. We haven't looked at the mice. That would be a great thing to do. And as far as other side effects emerging from the wheel of different functions, we've looked at a few of them. So for example, the canonical function is neuroid outgrowth. That's not affected. There are some other interactions of this particular channel of CRIM2 with other channels. Those aren't affected either. And I showed you CV2.2, which is a well-known target of this protein, is not affected by 194. So we're looking at that, but so far we don't see anything. Great. Thank you. Yes.
Hi. Very nice talk. I was just trying to think about it. Think about the peripheral versus the central question that you have, right? So you showed some nice data about the postsynaptic or no, presynaptic side having down regulation or down expression of NaV1.7, but just in terms of where NaV1.7 is on that on the neuron, like, do you see lower trafficking at the nerve terminal and also on the axons, and where exactly is the peripheral mechanism occurring?
Yeah, that's a great point. We haven't really examined that. We have a new construct in hand that was developed by Steve Waxman group that has a halo tag that we can now follow the trafficking. So with that, his group is looking at and seeing trafficking in the axon itself, particularly at the hillock. So we are trying to use those, but we haven't done those experiments yet.
Good morning. Congratulations for the work. I've been working on something similar, not the same target, but in small molecules development, and it's a rough path, and chronic pain is a really tough area to work on.
Absolutely.
But it's okay. My question is, have you performed safety studies in other systems, and which will be required for sure by the FDA? And my second question is, have you decided which of the whole spectrum of chronic pain will you go for and why?
Oh, very tough question. The last one's pretty tough, and often asked by my board to answer that one. The first question, in terms of looking at toxicity, we haven't done the GMP grade and then the tox studies in dogs, for example. We haven't done that yet, but we have done experiments. We've done tox evaluation in rodents, a dose escalation study for 10 days, and we don't see anything. A blinded pathologist to the treatments couldn't see anything in any of the tissues examined. And it was 100 milligram per kilogram over 10 days, escalating from 100 upwards. So early days, but we're still doing that. But yes, I need a lot of money to do the GMP grade and then testing in large animals. The second question is really sort of related to the first, because once it passes and it's safe in humans, then the question becomes, for example, could it be useful in CIPN? That's a pretty broad field that almost all chemotherapeutics have 70-80% incident pain. But then also it's very diverse. So I'm not sure that's the right one to go after. Most of the trials that Vertex, for example, or Pfizer have been doing have been in the space of post-herpetic neuralgia, for example. So something like that would be a condition we're thinking of. And being now in the College of Dentistry, we're thinking of more orofacial pain conditions that could be, because we have clinical, people running clinical trials there all the time. So that's something we're going after now. Thank you. Thank you.